Hi there guys, and welcome to another A-level biology video. Mr. Phillips, and today we're going to be looking at how we sample populations and do sampling of biodiversity. And I've put at the top here that this is really important because every year I find that students think they understand this well enough to answer exam questions, but they don't. So you really need to understand the process of how to sample and look at quadrats and how to take quadrats and get what you use the data for. And you also need to make sure you really do understand and so you can apply different conservation methods, different sampling methods, to make sure you don't lose easier marks on that exam, because a lot of the time students spend a lot of time revising like respiration, photosynthesis and biomolecules, and they overlook this, and that can be a very big dangerous mistake. Sampling in ecology is a big area of biology. Okay, so before we really get going onto this, let's just introduce some key terminology, just to remind you, which you should know from earlier on in the course. First of all, species, you should know, there are lots of different definitions of a species, but overall it's a group of individual organisms that have a similar appearance, anatomy, physiology, biochemistry. So you could say a group of similar organisms and just choose one of those. So a group of similar organisms with similar physiology that are able to reproduce to produce fertile offspring. And only 10% of species are currently known on the planet. And here are two examples of things that are produced that are infertile offspring. So when you produce two species, different species together. So you've got the liger, which is a lion and a tiger, and you've got a mule, which is a donkey and a horse, bred together, and they're infertile because they've been bred from different species. But obviously most of the time that is not physiologically possible. A habitat is a place where the organisms live, and a niche is a particular position or a function or a role that an organism plays in a particular ecosystem, depending on the resource it uses. And the ecosystem, remember, is all the abiotic and biotic factors in an area and how they interact with each other. So when we first of all look at this, just investigating populations in general, if we look at the world as a whole ecosystem, we think we've only discovered about 2 million of a potential 100 million species. We will know, the only way we'll ever know when we've discovered all of them is if the discovery rate drops. So what we're finding at the minute is you can go into areas of the rainforest or new like areas of the tropical seas, and you can find huge amounts of species very, very quickly and still discover them very quickly. It's only when we stop discovering new things that we'll think that we're near the limit of the total species we have. And the reason it's so slow is because it is actually really hard to go and discover different organisms. So for example, it could be inaccessibility to the ecosystem. So the rainforest, a lot of them we still can't get to. They might have very similar appearances, so we might think things look the same but actually are different species. Some organisms might be very solitary, and the males and females may not be obvious that they actually link together. And flowers sometimes are only identifiable when they actually produce flowers. So some plants, I should say, are only identifiable when they produce the flowers. And DNA and RNA methods are helping this now and speeding up the process and allowing us to see where things are actually related. So what is the actual Earth's biodiversity? Well, the WWF do an estimate every 2 years, but it's based on sample size. So basically, the larger the sample size, as we'll talk about later on, the more reliable your data, but it has to be balanced for the time available. So if you look at this, if you keep looking at increasing your sample size larger and larger, at point X, the number of new species in the sample levels off. So let's say you increase your sample size and you're finding 30 species constantly, and the same 30 species. That would be the time when you stop increasing your sample size. We've talked about biodiversity before in year one, but basically biodiversity is the range of species, organisms that are found, not just the numbers of each species. So you've got species richness and species evenness. So species richness is how much of species there are, and evenness how many different types of species are. And then we come on to this module really. So we're going to be looking at different things now, but we're going to be sort of looking at how we investigate it and focusing on a mixture of abundance and distribution. So the abundance is what the population is in an area. And you can do this by just counting the number in some cases, okay? But most likely you're going to look at the frequency or the percentage cover that you find in your quadrats. Now, in terms of doing this, it's much easier to do percentage cover than any of the others, because you could just literally look at a quadrat and go, oh, 70% there. Earlier on there was 20%. and compare the two. And we can use it to study distribution, which is where you find the species, and you can look at how distributions change. So the graph here that goes from green to blue shows us potential changes of reptile amphibian numbers due to climate change, and the green is where there's lots of them. So the blue is where there's lots of them. So we're expecting higher numbers in Europe because of the increased temperatures. So when we sample biodiversity, there's too many species often in an area for you to actually sample all of the plants and animals that are present that you're looking at. So here you've got an area of outstanding natural beauty in Wales called Unislas, and that is a couple of miles radius. So if you were sent there on a field trip for a day, you couldn't sample all the species in that area. So what you have to do is do a sample. You have to representative sample of the habitat, and you use random sampling. Okay, so depending on what you're studying would depend on the number, but it has to be, obviously the bigger the sample the better, but you have to be able to do it enough times to get enough data off it. So that's quite important to remember. And effectively what we have then is different sampling methods that we're able to use. So when we're looking at organisms, I've generally put plants there, but any organism that doesn't move a lot, so it can be sessile organisms like barnacles on a beach, things that don't move, or coral, which can be a mixture of different plants, animals and algae, that can be used through the quadrat and the transect, which we'll look at in a minute. But animals that move, we generally have to use a different method to obviously capture those animals. So quadrats are particularly useful for animals and plants that don't move, okay? And basically your average quadrat is just a metal frame, normally of a particular size. It's normally 0.5 meters squared, as the one pictured here is, and it's often divided into 100 subdivisions, which allows you to do percentage cover really easily, because if you're covering 4 boxes, it's 4%. If you're covering 50 boxes, it's 50% of the quadrat is covered in that by that organism. So how you'd use this is to do random sampling. So what you would need to do is you need to choose your area you're going to sample, okay, and then you would need to do samples to avoid doing bias. You need to do them randomly. So what you might do is take your field, as this diagram sort of shows here. You might split your field into random coordinates. So let's say you did A to F or something along here. Okay, so let's say you would do, let's just say A to Z because you know how big it is, and let's say this wide you did 1 to 10, so 1 to 20, let's say. You then pick your numbers from a hat or from a random number generator, and you'd pick like D12 and go and sample that area, F17 and go and sample that area, Z4 and go and sample that area, and that gives you a random sampling number amount, so you can go and do it without any bias in there. Okay, and effectively that's different ways of doing that random, but it's the idea of it being random, as you know, and you use those coordinates to go and find those areas. Now effectively then you could take, if you let's say did a field and the field was 100 meters squared and you managed to do 10 meters and you found a certain number of species, you could then times your answer by 10 to work out the overall population in that habitat. So there are two main methods of using quadrats. You could just use a quadrat, like I said, and just do random coordinates from the field, and that would be used effective as a counting method to work out the abundance of species. And basically you could do it random methods, as I said. You would then increase and multiply it by the total area to work out how many species were present. However, you might want to do a transect, and that is where you would basically, as the picture on the right suggests, you would do a tape measure. You put the tape measure along the field, and then as you did that, you then place a transect along the tape measure. Now there's two different ways. There's belt transects, which where you just place them constantly next to each other, so you do quadrats for the whole distance along the tape measure, or you might do what we call interrupted belt transects, where you leave a gap along, like you might leave a 5 meter gap between each transect, so you might discover a long period of time distribution changes. There are also people who use line transects, which is when it's just if it touches the tape measure that you include it. But most of the time I'd suggest you're going to be talking about using a belt transect, which is where you place quadrats along the tape measure. Now, when you place these down, as I said, so you generally use random sampling for your quadrats, which you do in a whole area, working the abundance of the population using the coordinates from a map. But for a belt transect, or like we've shown here, you, as we just said a moment ago, you might make them all so they're all touching each other, which would be called a just a general belt transect, or you might do an interrupted belt transect, which uses the idea of systematic sampling. So if you know you've only got a certain amount of time and you can only, let's say, do 20 transects in the time that you have, if you had 100 meters to study, you might do one every 5 meters, let's say, so you can cover it in the time and cover the whole distance in the time that you have to take the samples. Now, as I said earlier on, there's different ways of collecting the data. So one of the ways that I would tend to suggest is look at percentage cover, because it's easy to go to work out 100% cover all the different percentages. Sometimes they don't use numbers, they might link it to something, this one called the ACT4 scale. So you might say a species is abundant if it's got over 80%, let's say, common if it's, let's say, over 60%, frequent if it's over 40% of percentage cover, 20% is obvious, and rare if it's below 20%. And that isn't set of standard. You can use that for different things. You just need to display it at the start of your sample study. Now, you might use a quadrant and say, well, what happens if things go over it? Some people might say it depends where the roots are going into the ground, or you might say, well, if it crosses, say, this line of the quadrat and this line of the quadrat, we count it, but if it crosses the other lines, then we don't. Different ways that you could study these sort of things. Then there's something here which is a picture on the right, okay, and I'll come on to that in a second. This picture a bit better. This is called a point quadrat, and a point quadrat basically has 10 pins, and each one you'd count as 10%, and if the pin touches the plant, then you say it is there effectively. It's less prone to bias, because if the pin touches it, then you definitely say it's there, so it's less subjective. But you could have underestimations and overestimations. So if you've got one particularly large plant, it might only be one plant, but it might cover a couple of pins. And if it's less, really small organism, you might only pick up one, but there might be loads in the area. So it's just a bit that it can be affecting your validity of the results. So we sample animals. The problem is they're animals that move. They are motile, they move around a lot. So we might have to trap them, but that can disturb the habitat. So we need to be really careful with this to try and avoid it. So we use different things to try and sample animals. So we might use sweep netting, which you might use to do like insects in like fields, or butterflies or things like that. You could also obviously use a different type of net for aquatic animals. You may collect animals in something called a pitfall trap, so it might be used for different insects, and basically they're attracted in and then they fall in, and then you can collect them up and see what you've got. Or you might use something like moths, if you're doing that, something called a fulgurant tunnel, which basically is a light you'd leave at, say, in the middle of the woodland. The moths are attracted to the light, they get trapped in there as a result. And then this is called a putu, which you probably remember from primary school. And once you've got the, if you've got loads of leaflet and stuff, you can sort of suck the animals out to study them a bit closer. And different examples here are the same thing, just to show you what they actually look like, and obviously different mechanisms in there. But you don't need to know particularly that many, you just need to be able to apply it if you're given information about it. But what if the animal then is a lot larger? Well, you might use capture or release, or mark, release, recapture, as it's told about in your A-level books. So this idea is that what you would do is you'd capture a species with an appropriate method. So let's say if you were looking at snails or something, you might just go and find the snails and you'd paint a little point on them. If you're doing something like dolphins or sharks or something like this, you might bait the shark up to the boat and then tag it with an electronic tag, and then you obviously just release it back into the habitat. And that tag or that paint needs to be something that's not going to make the animal prone to predation. It's going to need to be something that isn't toxic, and it's going to need to last so you can see it when you need to come back. You then wait a certain amount of time, so it suggests in the book a week, but it depends what you're studying, and then you do a second sample in the same habitat, and then you use that to count the number of that you catch in the second time that been marked. So you work out your population size by doing the number that you got in the first sample So let's use some very, very easy numbers for this. Let's say you had 10 in the first sample, okay? You divide that, times that by the number in the second sample. So let's say we had 10 in the second sample, and let's say that 6 of them were marked. So what you would do is you would then do 10 times 10, which equals 100. Okay, and then the number marked in the second sample was 6. So then you do 100 divided by 6, which equals 16.6, and that would be your estimated population in that population. So here you go again, just a different way of saying it. So you've got population of a species, a mobile species. You do the first random sample and you mark a load of them. You then come back and you do a second sample, and in that second sample you work out how many have been marked. So in this example, the first sample they had 6 that have been that they've got and they've marked, and then in the second sample 7 were total caught, and only 2 of them had the marks on them. So you do 6 times 7 and then divide that by 2, which gives you 21. And it's only accuracy if the samples are large enough though. So obviously if you only do 10 in a pond with 1,000 fish in it, it doesn't make sense. You'd have to do it linking to the numbers in there. And just some extra information about this, the assumptions of this method is that the animals always come from the same population. So if animals move from one area to another, that can obviously be a problem. You must ensure the marking doesn't harm the animal or increase the risk of predation. You're assuming there's no migration in and out of the population, and there are no births or deaths during this time, because if you did it and then a week later the population crashed and you did it again, you can have a big disparity in your results. It could make you over predict or under predict. And larger animals such as whales can't actually be captured properly, so we might do skin samples and collect the DNA to build a database from that. And obviously more and more these days we are using for larger animals DNA databases. So that's been useful for you guys, and I'll see you next time.